Hi, everyone. Welcome. Today we have Carolin Gaiser, who's a postdoc at Department of Neuroscience and Child and Adolescent Psychiatry at Erasmus University Medical Center in Rotterdam, in the Netherlands. Carolyn did her PhD with Martin Friends, and where she was investigating human motor control and effective non-invasive brain stimulation. She also spent time as a visiting researcher in Ofer Dungeon's lab and Beersheba in Israel. Carolin's research focuses on the role of the cerebellum in motor adaptation and brain development. She's particularly interested in how differences in cerebellar development impact neurodevelopment health and how insights on individual differences in motor learning can contribute to rehab research. Thank you so much for presenting your work, Carolin.
Yeah, and thank you for the invitation. It's really a pleasure to be here and to present our work in this really cool seminar series with so many cerebellar experts also present. And today, of course, I'm also going to be talking about the cerebellum, and I'm going to be talking about cerebellar development in children and adolescents that we investigated in a large population-wide study with MRI data. And I'm sure that everyone that is attending this seminar is well aware of the importance of the cerebellum. But for a lot of people working on maybe the more clinical and psychiatric health side of neuroscience, the cerebellum still remains a little bit of a mystery and is really often neglected a bit. And that's despite the fact that we know that the cerebellum is important for social development and for cognition. And it even has been suggested as one of the key brain structures for some neurodevelopmental disorders. And one of those disorders that has shown quite some interesting findings recently with the cerebellum is, for example, autism. And when we look through these articles that I picked here, one thing that stands out a little bit is that these findings are still relatively recent, and also relatively recent, given that we've known about the cognitive side of the cerebellum for quite some time. So, for example, the cerebellar affective syndrome has been around for now almost 30 years. So, when we started off with this project that I'm going to be talking about today, we started this with the question of, given that we know the cerebellum is important in these neurodevelopmental disorders, and given that they often have their symptom onset in childhood and early adolescence, how does it actually develop during that time? And the way we wanted to find that out is by using the Generation R study. So the Generation R study is a very large prospective cohort that is based in Rotterdam in the Netherlands. So that's what the R in Generation R stands for. And it started almost 20 years ago with the inclusion of almost 10,000 children. And the interesting part here is that these participants that we study today, they have been enrolled in the study actually before they were even born. So the way this worked is that the Generation R study reached out to pregnant women at the time and asked them if they would like to participate in the study with their, at that point, still unborn child. The overall aim of Generation R is to find markers or correlates of typical growth and development, but maybe more importantly, atypical growth and health and disease. And the way the study is doing that is by gathering a large amount of data. So here is a little, a few kind of data examples that were taken very early on in the study. So we, for example, have prenatal measurements also of the brain with ultrasound measures. Here, for example, we also have some cerebellar measurements. We have medical data of not just the child, but also the parents. The parents filled in a whole battery of questionnaire throughout early life. And then once the kids were born, we did measurements on child behavior and child cognition. And this was done often very closely in collaboration with the GP of the families and also with the midwives. So after the kids turned 4, these measurements became a little bit more regular. with follow-up visits that were started then. And we have 4 follow-up visits up until now. So we have one after the age of 5, one after the age of 9, one after the age of 13. And we have recently finished the latest one, which was after the age of 17. And in these follow-up visits, you have to imagine that the participants are invited to the testing center, and they will be there for several hours doing several different tasks and undergoing different measurements. So that's anything from basic medical testing like blood pressure, weight, height, taking a blood sample, taking a urine sample, testing their vision, to a bit more focused measurements like, for example, grip strength or how high they can jump. They also always have the chance to talk with a psychologist in a structured interview. And we also have access to their medical records, and we know, for example, what kind of medication they have been taking or they are still taking. Maybe also interesting to mention is that the participants in the study are genotyped as well. And one thing I want to foreshadow already a little bit is that in this latest visit, we did a visual motor adaptation task. And by the end of the talk, I would like to talk a little bit about this one as well. But for the first part of this talk, I would like to come back to the imaging data. And at each of those follow-up visits, we also did an MRI scan. So I will be talking about structural MRI today, but maybe interesting to know is that we also have a resting state functional scan and a DTI scan at the same times. So we have around 1,000 scans in the first visit that we did, almost 4,000 in the second, and 3,500 in the third, which means that the results that I'm about to tell you about, they are based on these scans, which are around 8,500. So these scans have been around for quite some years. So I already mentioned the study has been going on for 20 years. And these scans have also been used quite extensively in a lot of neuroimaging research. But almost always, the way they have been used is with the cerebellum essentially cut out of the analyses. And that's not something that is unique to Generation R. This is actually what has happened in a lot of these large neuroimaging data resources. So our goal here was to do kind of the opposite, really, and that was to use the wealth of data that these study resources give, but this time to put the cerebellum at the center of the attention. And I mentioned just now that ignoring the cerebellum has a little bit of a history in neuroimaging research. And in a very recent paper by the Mello group, this is discussed in a very thorough and nice way. And they point out, for example, that they did a little meta-analysis where they looked at neuroimaging studies that say they do whole brain analyses, but in only about a third of those imaging studies, they actually include the cerebellum. So what is often happening is that whole brain and whole cortical analyses are used a little bit, are used as if they were the same thing in neuroimaging research a lot, which of course ignores that there are subcortical structures, not just the cerebellum, but also other subcortical structures that might be very important. So in this paper, they list out many different reasons of why that is happening. And I think it can be summarized in 2, let's say, overall problems. And the first one here is that, as already mentioned, the cerebellum is still known to be primarily a motor control and a motor coordination structure, which means that for a lot of researchers, they simply don't think it plays a big role in whatever research question they have. So this is one of the reasons why it's often ignored. But when it comes to neuroimaging research, I think another equally important reason are technical issues. One of them being the location of the cerebellum at the back of your brain means that when you set up your imaging sequence, you sometimes have to make decisions about what brain structures you want to focus on. And especially in these large imaging cohorts, people are the most interested in frontal areas. So if you want to have a good view of frontal regions in the cortex, then what happens is that you might cut away the cerebellum completely or just a part of the cerebellum. So if you only have a partial cerebellum, of course, it makes it very hard to include that into your analysis. Another reason is the very tightly packed cerebellar cortex, where you need a good resolution to calculate good outcome measures of it. And I borrowed here an image from the Spinoza Center in Amsterdam, where they show how different resolutions affect what we can see in the cerebellar cortex. And here they show a sub-millimeter resolution where you can really make out the folia in a nice way. However, that's not necessarily needed. So nowadays, most research scans have a 1 millimeter resolution, which is enough to get good measurements out of the cerebellum. And thankfully, this is becoming the standard, which makes cerebellar imaging research much more feasible. And last but not least, in the past, there has been a little bit of a lack of segmentation tools and analysis tools for the cerebellum. And that's really also important for these larger imaging studies where it's just not feasible to trace out individual lobules if you have thousands of scans. So having these algorithms available and also integrating them in the standard toolboxes that all neuroimaging researchers are using, I think that's really something that is happening a lot right now, and that makes cerebellar imaging research much more feasible and much more doable. So in the current project, we used two different kinds of atlases to parcellate the cerebellum. The first one being an anatomical atlas, the so-called maggot algorithm is what we use there. And this parcellates the cerebellum into its lobules, which you can see here in the different colors. And we used a slightly adjusted version of this atlas, where we also have vermal structures parcellated in here. The very neat thing about the maggot atlas, or the maggot algorithm, I should say, is that it allows you to tailor these the algorithm towards your own cohort. And that's especially interesting here because we have a developing cohort with young children and adolescents where often these algorithms work very well if you have an adult brain, but maybe less so if you have a developing cohort. So here we could really make use of this, which also means, however, that it's computationally quite intense. The other atlas that we used is a functional atlas by the group of Jörn Dietrichsen, where the cerebellum is parcellated in 10 functional regions that, again, you can see here in different colors. And there is, for example, regions that relate to motor function here in the anterior cerebellum with an inferior representation here as well. And then we have the large cerebellar hemispheres where we can find a lot of cognitive functions such as arithmetics, attention, and language. One thing I want to point out here is that on this flat map, you can see these dotted lines, which show you the lobular boundaries, which you would get from an anatomical segmentation. And what is shown very nicely on this flat map is that these functional regions are really not contained well in lobular boundaries. So we really wanted to have both of these atlases available in our cohort because we think of it as looking at the cerebellum in two different ways. So these are the regions of interest that we investigate. And when we started this project, we wanted to just plot them over time and see how they develop. And very often when we would do that with just the raw outcomes, let's say volume in a certain region, we would see a trend like this with lower values at the first measurement wave that we have and higher volumes or any kind of outcomes at the second and third measurement wave, which kind of suggests this curvilinear trajectory that is kind of reaching a ceiling maybe by the age of 13, 14. Now, One thing I haven't mentioned yet is that in our study, we updated the scanner after the first wave, which means that all of these measurements are done on a different scanner than the subsequent measurements. This means for our outcomes um, that they could really differ. So what happens is when you put the same person in two different kind of scanners, the kind of outcomes that you would calculate from the scans that you get, they can vary by quite a bit. And this is something that's happening a lot, especially in these long ongoing research studies, where at some point you either update a whole scanner, or maybe a head coil breaks and you have to change this, or at the very least you make you have a software update. But this is all things that also influence and confound your data. So we wanted to find a way to correct for this, because we really cannot tell by just the raw outcomes if we would have seen more of a linear increase like this if they were all taken on the same scanner, or maybe something completely different, which, for example, in the case of volume, is unlikely. But the truth is, with just the raw data like this, we couldn't really tell what the true trajectory is. And the way we try to solve this is by using something that is becoming increasingly more popular in neuroimaging research, which is called normative models. So normative models work very similar to what we know from growth charts in pediatric medicine, where you can check, given a covariate, so for example, age, how for example, part of your brain should develop. So let's say you have a 9-year-old. How large should the cerebellum be? And at what point is it maybe too large or too small? And these normative models can be trained on large data. And what they do is they also take into account that the information, the source data, is coming from different sites or different scanners. So this way you can calculate the differences between scanners and can essentially subtract that from your data. Another very nice advantage here is that this allows you to derive subject level statistics. So after running the normative models, you can tell for each individual how far off certain areas in the brain are from the expected norm. So this removes the need for a case control study. Another very nice feature here is that it allows you to share your results without needing to share the data that these models are based on. And that's often the case when you have large data sets that, for example, include patient data, that you're just not able and allowed to share such sensitive information. But here you can give on these results, these normative models that have all the information from all the 1,000 scans without including the source data. And it can even be adjusted and updated by new data. If you want to, for example, use it in a smaller cohort, you can adapt these normative models and you can increase your power by being able to train your data on this prior information that is based on thousands of subjects. So this was how we set up the study. And now for the I think most interesting part of this, the results of what we found. I will start with the results in the anatomical atlas. So what we see here is how much the cerebellum grows in the first row in males, and in the second one in females. And the color here shows growth in our age range, which is from 6 to 17. So basically, the more red the color is, the more increases here in volume do we see, the steeper the slope is. And the first kind of obvious thing, but I'm still going to highlight it, is that all of the lobules in the cerebellum are still increasing in size. So this is not too surprising, given that we have a developing cohort of kids. But another thing, if we look a little bit closer here and go along the cerebellar cortex, is that we find that there seems to be more growth happening the more posterior we go along the cerebellar outlines. And we can look at this also in terms of trajectories by taking an example lobule in the anterior cerebellum, so here lobule 5, and we see that volume still increases, but not as much as, for example, in a posterior lobule here, CRUS 2, and we see the most increases actually in the corpus medullare, in the white matter tract of the cerebellum. So this is quite interesting, but then we, of course, wanted to see how this relates to possible underlying functions in the cerebellum. And here I brought up the functional atlas again that we used, just for a little reminder that the motor parcels, here parcels 1 and 2, are mostly located on the anterior cerebellum with a small posterior representation here as well. And if we check how volume in these areas develop over time, we see again that the whole cerebellum is growing over time, but there seems to be this distinction between motor parcels and the more cognitively related parcels. We can look at this not just in terms of volume, but also in gray matter density and white matter density, where we know that gray matter density decreases in that age range and white matter density increases. But we see again this distinction between motor and non-motor areas. If we look at this with trajectories, so here this is the left motor parcels, we again see that really not much is going on anymore in gray matter density or white matter density. But if we take a posterior parcel, here one that is involved in divided attention, we still see decreases from 6 to 17, and we also still see increases in white matter density from 6 to 17. So from this, we concluded that there seems to be an anterior to posterior growth gradient, and that seems to have something to do with the underlying cerebellar functions. Interestingly, this is very similar to what we know from the cerebrum as well, where previously it has been shown that these more basic functions, like for example, the somatosensory cortex, these are the areas that mature the earliest in the cerebral cortex. And areas like, for example, frontal areas or more temporal areas, that they have a prolonged maturation. So our idea here and our interpretation of this would be that the cerebellum and the cortex, they seem to correspond in a very similar way, which again, given how highly they're interconnected, doesn't seem too surprising, but it's kind of nice that there's this correspondence. What we then also did is we wanted to display our results in the Little Brain Toolbox. And the Little Brain Toolbox lets you visualize neuroimaging results of the cerebellum along 2 gradients that have been found previously. And these are principal functional gradients of the cerebellum, where the first one that is shown here on the y-axis pertains to motor function. So here, low values on this axis are areas that are involved in motor tasks. And then here, these would be unfocused areas, like for example, the default mode network. This would be one gradient. And then the second gradient, where this is, which the Little Brain Toolbox uses, pertains to cognitive load, where one end of the cognitive load scale is kind of the extremes of gradient 1, so motor and default areas. And the other end of the cognitive load gradient pertains to, for example, working memory tasks or frontal parietal networks that are very high in cognitive load. And this toolbox lets you plot voxel data of the cerebellum. So each and every point here in the scatter plot represents one voxel in the cerebellum that is then located in these different functional areas. And if we plot the growth that I showed before in this system, then what we can see here is that more growth can be seen as indicated by this red color in areas that seem to be high on cognitive load, where we see less growth still happening in areas that are, for example, responsible for motor behavior or in the default mode network. So with this, we concluded that this growth gradient that we see might actually have something to do with cognitive load, with areas that are high in cognitive load needing a longer time to mature than areas that are low in cognitive load. So before I continue on this, I just quickly wanted to summarize the main results that I just talked about. So we found a growth gradient in the cerebellum, which seems to go from anterior to posterior. This growth has a lot of similarities with what we know from the cerebral cortex, and it might pertain to the underlying functions in the cerebellum and potentially also to cognitive load. So this is what we found, but I thought it would also be very nice to show an example of how these growth trajectories can now actually be used in, for example, a clinical application. So here I'm going to show you data of children in our cohort that are born very preterm. So very preterm means that they were born before the 28 weeks. And I will show you two different kids that are born very preterm. And the reason why I chose preterm birth is because it really highlights the advantage of having these single individual level deviations. Because to give you an idea of how often that happens in our cohort, where we have thousands of kids, I think if I remember correctly, we have 6 kids that are born very preterm. So if you have only so little kids in one clinical phenotype, case control studies get very difficult. So starting with the first kid here, we see how the cerebellum is different at the age of 9 for this kid that is born very preterm. And what we can see in terms of volume, gray matter density, and also white matter density is that actually not much is going on here. It's hard to tell by the color bar because everything is kind of the same shade of yellow, maybe a little bit blue, but everything we see here is within one standard deviation of what we would expect from typically developing kids. Now, if we look at a second kid that falls under the same umbrella of very preterm birth, but is only born a few days earlier, we see a very different picture. So we see, for example, that at the age of 9, this child has much lower volumes in the cerebellum than what we would expect. And we see a kind of a mixed picture in terms of gray matter density and white matter density. But especially on the right hemisphere, we have lower gray matter density than what we would expect from a typically developing kid at the same age, and higher white matter density from a kid at the same age. So what this shows us is that there's a lot of heterogeneity in this group, as is the case with many different disorders. And especially if you then not have the power and numbers for a proper setup case control study, it's very advantageous to have these normative models where you can really look into deviations on a single subject level. So for people that would like to use these growth trajectories, they are open access. They can be downloaded either from this GitHub link that I show over here, or they're also available on the PCN toolkit and the PCN portal. And PCN stands for Predictive Clinical Neuroscience Group, which is the group led by André Marquand, located in Nijmegen in the Netherlands. And they're really the creators, the makers of this normative imaging approach that we used here. And there's a very neat feature on this PCN portal that they have, which is that you can, given that you have the same regions of interest as, for example, I showed previously, you can upload an Excel file with your data, and the computations for this normative modeling will be done in the background, and you will receive an email with individual level deviation scores. So this way you can receive these scores without ever needing to touch any kind of code or adjusting anything, and it makes it very, very applicable. If you would like to dig into the code a little bit and adjust these models yourself, we also wrote a little methods paper where we give recommendations in, for example, modeling settings, where you can adapt new data into existing normative models. And we do this here with an example of cortical data, but of course the same can apply for cerebellar data as well. So I also wanted to take the opportunity here to talk a little bit about future plans and what we would like to do next with these cerebellar imaging trajectories. And one thing we would like to do next is to connect it to behavior. And I already foreshadowed a little bit that we did a visual motor adaptation task in the latest generation R measurement wave. And it feels a little bit redundant to explain this here because I think there's people in the audience that know this task a lot better than I do. But here I go, I will still do it. So what we did is we seated participants in front of a table where they operate a handle underneath the table with their right hand. And we obstruct their view so they cannot see their arm or the handle underneath the table. Instead, we project a visual cue on top of the table and simply instruct them to move this visual cue using the handle to a target that is 10 centimeters in front of them. So after some time, we introduce a very small visual perturbation. We do that in a stepwise fashion of always 2 degrees, and people learn to adapt to that in a trial-to-trial fashion. So this standard paradigm allows us to quantify individualized parameters for cerebellar learning in the generation R cohort. And we were able to do that in over 2,500 participants. And the way we're doing that is by fitting a state space model of trial-to-trial learning. And the individual parameters that we get out of this is a learning rate, but also noise parameters. So for example, planning noise, which is thought to represent exploration behavior, and execution noise that is coming from more the periphery, so the muscles, for example. And on the left here, you see an example of 2 participants that were doing this task. And what you can see in blue is a control subject, a healthy control subject, and in red is a patient that is very high on the autism spectrum and also has ADHD. And what you can see here is that they both are able to perform this task. They can both adapt their movements to our perturbation, but the behavior is quite a bit different. And this is also reflected in these individualized parameters that we can calculate. So they both learn the task, but this patient is really characterized by a lot of execution noise, by a lot of variability in his movements. A colleague of mine, Rick van der Vliet, has also shown that these noise parameters relate differently to how well people learn. With planning noise, so this exploration behavior, usually correlating positively with how well people learn, and execution noise relating negatively to how well people learn. So what we would like to do here in the Generation R study is to use this rich data set, all these measurements that we have, and basically figure out what makes a good motor learner. Is there a subgroup of people that do really well in this task? And if so, what are the common characteristics here? And even more importantly, how are the people or who are the people that are not doing as well in this task? And what do they have in common? So we can maybe tackle these issues to help them with motor learning, which might be interesting in a rehabilitation setting, for example, after you've had a stroke, if you need to relearn movements again. Another kind of obvious question thinking about that this task we know is very much dependent on the cerebellum is to connect what I was talking about in the beginning of the task, in the beginning of my talk with cerebellar growth to the behavior later in life. So how is the early development of the cerebellum related to how well they're doing in the cerebellar dependent motor task later in life. And lastly, the Generation R study is ongoing. So there's going to be a new measurement wave that is probably going to start this year still. And right now we're very much in the brainstorming phase of thinking about and piloting certain things for the upcoming wave, where we again would like to do a visual motor adaptation paradigm. But this time we would like to influence and possibly improve learning in these children. And we are thinking about doing a reward and potentially also a punishment paradigm here to see how this influences participants and maybe a certain subgroup of participants as well. But I kind of just wanted to put that out there and take advantage of the fact that there's many people present with a lot of knowledge on this. So if anyone has any ideas or would like to put some considerations forward here, that would be very interesting, and I would be happy to hear them. So to summarize quickly what I've talked about today. So in the beginning of the talk, I showed open access cerebellar growth models, which correspond to what we know in terms of maturation from the cerebral cortex, and also correspond to potentially underlying functions in the cerebellum. We used a normative modeling approach, which makes it possible to investigate deviations on a single subject level, which is very useful if you have rare phenotypes or if you have phenotypes that are very heterogeneous. And for the future, we have some project plans where we want to connect this to motor behavior, but also actually we would like to also use these models to investigate clinical phenotypes more. Unfortunately, I didn't have time to talk about all of this. And we also are working on extending these models by including more imaging modalities and to also extend the age range. So right now they go from 6 to 17, but ultimately we would like to have these models for the whole lifespan. So with that, I would like to thank some people. First and foremost, the Generation R study and the participants there, who continue to come to the testing center and spend time and effort, which makes this study possible. I would also like to thank collaborators that helped me with working on these projects, and a special thanks to my two PIs, Marte Frenz and Ryan Mützel. And of course, thank you for listening.
Thank you so much, Carolin. That was just wonderful. What a beautiful talk. Please go ahead and raise your hand, and I will call on you if you have questions for Carolin. Carolin, maybe I will start by asking you sort of a dumb question about your results. When you were showing data associated with white and gray matter density and the changes that had taken place in with those two measures, I thought that they were often inversely correlated, meaning that if you found something above the mean in white matter, the density, the opposite was true for gray matter.
Yes.
First of all, is that true? And if it's true, I don't quite understand what does that mean?
Yeah, so that's a good question. So that goes back to how these gray matter densities and white matter densities are calculated. So they are a probability of a voxel either being gray matter, white matter, or cerebral spinal fluid. So they are related. It's not completely opposite all the time, because there's also a chance they could be cerebral spinal fluid. But they're basically probability values that range from 0 to 1, 0 meaning in terms of gray matter density, it's very, very unlikely that there is any gray matter present. 1 meaning this voxel is completely gray matter. So it does make sense that this changes kind of in opposite relation, yeah.
Jessica, you've raised your hand. Please go ahead.
Yeah, so Carolin, thank you. This is a really, really wonderful. Can you hear me okay? I just want to make sure.
Yeah.
Okay, great.
Yeah, I see.
Like, this was a really wonderful talk. I really love the way you approach this data set and how you analyze this. I was thinking about this. So I noticed you separated out your males and females. Have you considered the influence of pubertal timing, thinking about recognizing, especially in females, there's a high level of receptors for 17 beta estradiol and progesterone, at least in the adult female cerebellum, how this might come into play when you're thinking about these growth patterns and trajectories?
Yeah, that's a very good point. So we haven't done that yet, but the reason for not having included more covariates, because there could have been a few more things that we could have considered here. So puberty onset could be one of them. Also certain clinical phenotypes could be others. The reason why we haven't done that yet, but potentially in future projects we might consider it, is because we wanted to develop kind of a basic model that is useful to a broad audience. And a lot of times you don't necessarily have this data for puberty onset, which you would then have to feed into these trajectories as well. But it's a very good point. So there, of course, then trajectories would change slightly. Yeah.
Thank you.
We have a question by iPhone, but I don't know who that is. So if you want to go ahead and introduce yourself and ask your question.
Hi, good morning. Sorry, I'm having some technical difficulties, but I can go ahead and ask what I asked in the chat. So great and amazing work. I was curious if you tried modeling quadratic age trajectories for your age range. And if so, what did you find? And did you like formally compare the models?
Yes. Yeah, thank you. I didn't have time to go into this, but in the paper we described that. So we fitted two different kinds of models. We fitted just a basic linear model and also a B-spline model, which would then be able to capture, for example, quadratic changes, but also a bit more complex changes. And what we found was that at least from 6 to 17, the just linear models fitted much better. So we compared the two with a leave one out cross validation and found that almost for all regions of interest, there are some functional ones where maybe a quadratic one would fit better, but for almost all of the regions, just a simple linear model was the one that fit the data the best. Yeah.
That's so interesting. And what you had mentioned this earlier, which covariates did you include in the growth trajectory models? You did ETIV, you didn't do biological sex because you did separate models for the sexes, right? But did you include anything else in the actual models?
No, so we tried to keep the model very straightforward. Our main predictor was of course age, and then now that is I don't know how you want to use the terminology here, but we corrected, so we stratified basically for sex, for biological sex, yeah. And the other covariate that we included was then, of course, the fact that data came from different scanners. But other than that, we didn't include any other covariate.
So you had a dummy categorical variable for scanner type?
Yeah, exactly.
And you did not control for head size?
We did not control for head size, no, but then it comes to the question of, so if you control for head size, we didn't test that, but I agree there would be a good sensitivity analysis to add to this. Some of the changes of interest would get eaten up by the fact that just the head is growing over time, right? So we decided to keep it to keep the models without, for example, an ETIV or a total brain volume. Yeah.
Did anybody push back on that during your review?
Funny enough, no. But we would have been open to just run it also with ETIV, right? So that then the interpretation of your trajectories basically changes, right?
Let me go ahead and on to the next question. Thank you so much. Yi Chen, you have your hand up.
Yep. Thank you for carrying your wonderful talk. I'm Yi Chen from Columbia. I have two questions. The first is, when you show the chart 2, and I see there's a right side and left side difference. So, I mean, it's very interesting to see. I mean, right side and left side when they developing their cerebellum volumes. And the second question is, you mentioned that you have resting and DTI sequence, and how do you think resting or DTI sequence is more sensitive compared to the structural imagings?
Yeah. Yeah, thanks for these questions. So the first one about lateralization of the growth, it's a very interesting one, and it's one that I want to go after a little bit more because there is also data on the cortex, of course, that there is some lateralization going on in terms of maturation, but it's kind of hard to pinpoint exactly what is happening there and if this is if this is what this is really telling us in the end. So I agree, it's interesting. I'm afraid I don't have a very clear interpretation of that at the moment. And when it comes to the other modalities that we have, that's one of the future things that we want to look into. So especially the DTI data, I think here could be could hold a lot of valuable information about how the cerebrum and the cerebellum mature in unison, how they mature together over time. So this is one of the next steps that we wanna tackle. Yeah.
Thank you, Cheng. Thank you so much. Will, go ahead. Please ask your question.
Thank you very much for your very insightful talk. I think it's beautiful work in terms of the large data set, developmental and imaging data sets. And I see the implications for the motor domain. I wonder what I can learn from your work on cerebellar contributions to cognition or cerebellar connectivity contributions to cognition. What can you try to help me to understand?
Yeah, I think that's a good point. So what I've shown in the first part of my talk is really descriptive, right? So it's development over time and just how do these areas change and how do they change relative to each other. And there seems to be this difference in terms of cognition, where areas that are more cognitively demanding seem to need more time to mature in terms of volume and gray and white matter density. So the takeaway message here for cognition is really just that this seems to be more demanding, and potentially the brain needs longer to figure that out, and more there is still happening in childhood and adolescence, whereas with motor behavior, with a lot of that and a lot of that development, we might just be done, let's say, after the age of 10, 11, 12, or very little changes are still happening there. If we want to go a little bit further with this, I think the next step there is really to combine the cerebrum into this, because there is a lot of information about cognition and development on the cerebral cortex, and I think we shouldn't make the mistake of what the cerebral cortex people have done in the past of only looking at the cerebral cortex, and we now only looking at the cerebellum. But both of them really have to be studied in a combined way, and I think then we can answer a lot of these questions in a lot more complete and sustainable way. Yeah.
Thank you and good luck.
Thank you.
Amy, please go ahead.
Oh, that was a great talk, Carolin. I don't know if you can hear me well. Yeah. Excellent, really terrific. I'm curious about the differences between right and left hemisphere and development. It went by a little fast, but what are you seeing there? It looked like there was a difference.
Yeah.
There we go.
Yeah, so that's indeed, again, the kind of lateralization that seems to be happening here. So what we see here is that it seems like the right hemisphere is not changing as much anymore as the left hemisphere. As I mentioned previously, I find it hard to really pinpoint why that is happening. Also, if that might be, yeah, there's a little bit of a difference between males and females here as well. So the first row that I show here are males. The second one are females, and it looks like in females here, the maturation of the right hemisphere has already finished, is already complete. So maybe there is something to it, but I think that this would have to be tested properly in a separate project, because all we're doing here is really just descriptives of what we see in the data. Yeah.
Right, right. I was going to also just share, we have a couple of papers coming out on growth that have growth, one on adaptation. It's a growth chart from age 3 to 17 of adaptation ability that's in press. I can send you a copy of it. It's about 600 kids, but it's an at-home game. You can just push it out to them, and it might be useful in thinking about future tasks.
Oh, that's really cool. So it's a like tablet-based measurement, or
you can do tablet, mouse. You can do, trackpad. We have a reinforcement game as well. We haven't published the growth curves yet, but that paper's on bioRxiv, and it's back at eLife. So some of these games may be useful because you can send them home to people. And we collected all the data from all over the US at home. So
that's very interesting. Yeah, that
I can send you preprint. Yeah.
I would love to read that. Yes, thanks a lot.
Thank you.
Nikolaj, please go ahead.
Okay, can you hear me now?
Yes.
Yep.
Okay, cool. Well, there's something wrong with my, okay. So I have two questions. First of all, I think it's very, very interesting to see the data on cerebellum coming from generation R, because you will see a lot of stuff on development and maturation of the white matter, which I think is fascinating. But I have two simple questions. One is regarding the super sampling. So in current situation, we are trying to extract as much as information is available from 1 millimeter isotropic resolution. And I think the problem is that we have a lot of volume averaging. So what do you think would be the ideal resolution if you had to pick one? That's number one. And number two, I mean, like realistic. And the second question is, did you notice that the currently available skull stripping protocols are failing quite often on younger kids, and we should have cerebellum specific skull stripping effort?
Yeah, thank you very much for these interesting questions. I think you started off saying that these are simple questions, but especially the first one with what is the ideal resolution for cerebellar imaging, I find quite hard to answer, to be honest, because it's always a trade-off. Preferably, you would have like 0.2 millimeter resolution to look into all the folias of the cerebellum. But that's just not feasible, especially if you would like to look into large population-wide cohorts, and especially if you want to look into children as well, who would have to lay still in the scanner for a long time in a high field MRI scanner. So I do think we can do quite a lot with 1 millimeter already. That is not to say that that's the ideal resolution. I think the ideal resolution is the resolution that you can do in an appropriate amount of time to get the best amount of data out of it. It's a very vague answer.
No, I totally get you. It's in other words, pick up the resolution that it's achievable.
Yeah. That also is practical for researchers to do. Yeah. And then we can still do these specialized studies in 7 and 14 Tesla scanners, where we really look into the details and really try to have these beautiful images of the cerebellar cortex and try to see what's going on. But I think if we look at these larger studies, that's just not feasible, at least not at the moment.
So stick with what we have, in other words.
Yeah, make the best out of what we have, I would say.
Okay.
And the
Skull
second,
stripping.
it was about skull. Yes, thank you. So what we have done is we used the standard pipeline of imaging processing for skull stripping that was not dedicated for the cerebellum. So that just the normal ends and free surfer pipelines that actually worked quite well in our case. Even with the younger children, we sometimes had problems due to motion artifacts, but in these cases, really the data was also of lower quality. So even if the skull scripting would have worked better, I doubt that we could have used the data and corrected for the motion in a well enough way. Yeah.
Okay, thank you.
Thank you so much. Looks like there's one more question. Go ahead, iPhone.
Oh, thank you. Yeah, I wasn't sure if it was me. I was curious about the maggot pipeline, parcellation pipeline choice. Did you try any other ones like Acapulco or Sooth Atlas? Why did you choose maggot? And also, do they only take a few subjects on their website? Is that how you did it? Or did you do something else to process all of your data set? Thank you so much.
Yeah, thank you. I really like that question because that's where we're gonna go at next, because Acapulco seems to be one of the cerebellar segmentation algorithms that's gonna be used a lot, and that already is used quite a bit. So we want to rerun the models with Acapulco as well, and maybe also CerebNet and some of the others that are out there. But the reason why we chose for maggot here is because we really wanted to have a way to adjust this to a developing population. And the way this works in the maggot algorithm is that you have these 5 atlases, I think that you mentioned, from the maggot group, where they have manually segmented the lobules of the cerebellum. And then you use a number of template images. In our case, we used per measurement wave 7 template images, which makes 21 because we have 3 measurement time points. And they were used to adjust these 5 manually segmented atlases of the maggot brain. And they were then propagated to the rest of our sample, so that these template images could really be used to tailor the algorithm a little bit to our images.
Thank you. That makes a lot of sense. Yeah. And good luck, by the way, with the Acapulco.
Yeah, thanks.
Carolin, that was wonderful. Thank you so much. And thank you, everyone.
Thank you.
Bye-bye.